Well, welcome, everybody. My name is Daniel Rothenberg. I co-direct the Center on the Future of War, which links the institution we're at today, obviously, New America, with Arizona State University based out in Arizona, of course, although also with an office here. So we're thrilled to have such an exciting panel here with the release of a report that's available outside if you haven't already picked one up. So very briefly, I'm just going to introduce all of our panelists, pass things over to Jesse to present the plan for the event, and then there'll be presentations from all the panels. So Jesse Kirkpatrick is an International Security Fellow here at New America and works quite a lot with ASU as well. He's a research assistant professor and interim director of the Institute for Philosophy and Public Policy at George Mason University. And he's a political military analyst for Johns Hopkins University, the Applied Physics Laboratory, and an expert on the ethics of peace and security with an emphasis on emerging technologies. Gregory Koblentz is an associate professor in the Schar School of Policy and Government and the director of the Biodefense graduate program at George Mason University. He's also an associate faculty at the Center for Security Policy Studies at George Mason and the member of the Scientist Working Group on Chemical and Biological Weapons at the Center for Arms Control and Non-Proliferation in Washington. His research and teaching focus on international security and weapons of mass destruction. Dr. Megan Palmer is a senior research scholar at the Center for International Security and Cooperation at Stanford University. She leads a research program on the governance of biotechnology development with a focus on how security is conceived of and managed. Her current projects focus on assessing strategies for governing dual-use research, analyzing the international diffusion of biosafety and biosecurity norms and practices, and understanding the security implications of alternative technology design decisions. Edward Perello is the principal researcher for Arcurity, a boutique consulting firm conducting research on public policy challenges in synthetic biology, conservation, biotech, and biosecurity. He's also a research fellow at George Mason University, where he works on security policy related to genome editing tools. His research interests include the oversight of human genome editing, state and non-state actor development of biological capabilities, and the application of synthetic biology to ecological challenges. And finally, we have David Relman, who's the Thomas C. and Joan M. Merrigan Professor of Medicine and Microbiology and Immunology at Stanford University. And he's the Chief of Infectious Diseases at the Veterans Affairs in Palo Alto Healthcare System. He's also a Senior Fellow at the Freeman Spogli Institute for International Studies, where he directs the Biosecurity Initiative. And he's involved in a variety of different high-level initiatives, including Vice Chair of the National Research Council Committee that reviewed the science performance of the FBI, the FBI anthrax letters, if you remember, and the chair of the Forum on Microbiological Threats, and a member of the Committee on Science, Technology, and Law, and currently a member of the Intelligence Community Studies Board, all of which are part of the U.S. National Academies of Science. And he was elected to the National Academy of Medicine in 2011. So, thank you, Jesse.
Great. Well, thank you, Daniel, and thank you, everyone, for being here. It's always a pleasure to be at New America and to spend time with Daniel in particular. So we're going to try to keep our remarks relatively brief today. We're going to present the outcomes, findings, methodology, and structure of the study that we've run over the course of 2 years. And we're each going to be presenting in turn, and I'm going to be starting off with giving you just a little bit of a brief overview and introduction to the study, the sort of structure and design, and the motivation. So, sorry, I need to just. So the purpose of this project was to really kind of dig deep on questions related to risk, security, and genome editing. We decided to focus in particular on a type of genome editing, CRISPR, which many of you are likely familiar with, and Eddie will be giving an overview for those of you who aren't about how the technology works and why it's important. We want to focus on the security dimensions for a number of reasons. One is that these dimensions are that they're complex, that they're studied often, but what we found is that we wanted to craft a study that had a team of experts who really cut across disciplines. So we had a series of stakeholders from backgrounds including science and technology, the humanities, particularly ethics and bioethics, security, policy, social sciences, and technology. And the reason that we wanted to do so is that we wanted to be able to speak to a wide audience, engage a range of stakeholders, and we'll explain in a little while why that's important. And we want to contribute to the suite of studies that already exist in this space. David's going to be speaking a little bit about that in a few moments, so I won't say too much more. So here's the project team. We've already been introduced, so we're live and in person, and you know here. But a little bit about the study process. As I mentioned, this was a 2-year study. The backbone of it was really three workshops, in-person, invitation-only workshops. The first one occurred at George Mason University, and one at Stanford, another one at George Mason. Underpinning these workshops were the commissioning of several issue briefs. So here are the project contributors, many of whom wrote these issue briefs that you see here. And we commissioned a number of issue briefs on a variety of topics. I won't go into them in detail. They're behind me, as you can see. But we reached out to experts and asked them to author these issue briefs so that we could do a deep dive on various areas related to genome editing and security in particular. To bookend each of these issue briefs, we also authored 2 working papers. One that kind of gave a lay of the land of risk and security assessments, kind of state of the art or state of the field, I should say. The second one was looking at frameworks for policy and governance. So a little bit about to give you some foreshadowing is that we really sort of grouped our takeaways into kind of 2 major categories that speak to the methodology and the design of our study approach. And the first is using CRISPR, genome editing, and CRISPR in particular as a lens. And that's a lens to view the complexity of this emerging security landscape. And the second is to use genome editing as also as a focus. So we wanted to use it as both a focus and a lens, a focus to drill down onto these complex security issues, and then as a lens to kind of zoom out and see some of the broader takeaways or broader dimensions of biosecurity in general. So one of the real questions that we asked 2 questions. The first was, how is genome editing in particular affecting this space? The second was, how is it affecting biosecurity more broadly? So a number of our colleagues, including David, have been on a host of studies, panels, and have a long history, not too long, but a long history of experience in this space. So I want to hand it over to David to put the project in a little bit of context.
Thank you. I'm going to make just a few brief comments, but I hope you all know that we're happy to return to any of these points in more detail afterwards. But just a few points about challenges, about current and ongoing efforts, and about the contributions of this study. First, on the topic of challenges, I think one can easily imagine that there are several. First, there are a multitude of advances in science and technology that might be worthy of this sort of focus, as well as a multitude of risks. And around each of these, there is a great deal of uncertainty about likelihoods and consequences. And that's true for the benefits as well as the risks for each of these possible areas of study. There's also, in our estimation, a dearth of adequate landscaping effort to stand back and look at this array of possible advances worthy of study and think about, where does one begin? How do we prioritize? Are there any that deserve much greater attention than others? And finally, there's the challenge of geography, of cultural variation in the way in which humans, populations of humans, look at any of these issues and think about where they might have concerns, where they might see unusual opportunities, and how to balance those two. So those are some of the challenges that we thought about and try to address. On the topic of current and ongoing efforts, all of you know well of many of these studies that are highlighted here in little images. There are plenty of others as well, and I think each of these studies had important things to contribute to the conversation about the topic at hand here. When we looked at these, and we did in a somewhat deliberate way, we realized that there was, in many of these cases, unusual focus on a particular problem to the exclusion of others, and involvement of a particular set of participants in the study to the exclusion of others, such that you might imagine the effort to be understandably but necessarily limited by both of those things, so scope and kinds of participants. And finally, there's been relatively little follow-through or follow-up to any of these previous studies. We can't know that we will have engendered that here, but we address this issue of how do we create a process rather than a product that has lasting and ongoing meaning and activity. And so that would be one of the points we would make about the contributions that we hope to make with this report, that in essence, we want as much to use this particular technological advance, as illustrative of how a group of diverse people might sit down and think through this or some other problem, think about scenarios, work through scenarios, and by doing so, realize the realities of what it will take to do something useful, and then make that an ongoing process. And so think about issues that some call adaptive governance mechanisms or regimes that iterate and think through how to better the kinds of actions that have been taken before. So I will hand this on.
Thanks a lot. So I want to walk you guys through genome engineering technology and wetware, the tools that are actually used inside cells, so you can get an understanding and an appreciation of what's going on at the molecular level, as well as what's going on at the application level, why are people using it, and then a little bit higher up at the industrial and sectorial level. So what you see behind me is a schematic representation of what's called the central dogma of biology, effectively that DNA, the thing that we care about most in this discussion at least, is turned into RNA and then eventually into protein. And when we're talking about genome engineering, what we're talking about is editing DNA in order to modify the proteins at the resultant end of this process. So why would we want to edit a protein? Well, proteins make up a huge chunk of our bodies, and cells that we want to experiment with, they are the hormones, they are the digestive proteins, the enzymes, and so on and so forth. They are essential for organisms to function. It's very difficult to modify proteins directly, but if you can modify the source of the protein, the DNA itself, then you can make very specific changes in these proteins. You can change the function of the organism at a very precise level. So what I'm about to show you next is how this occurs at the molecular level using a genome engineering tool called CRISPR. And I'm going to show you a video which comes from the McGovern Brain Institute. We're starting a little bit late in the process where the CRISPR molecule has already been introduced to a cell. So here we go. This is the Cas9. This is the nuclease protein which is capable of cutting DNA. And the blue strand, that double-stranded molecule, that's the genomic DNA. It's unwound by the Cas9, and that red element is a programmable guide. You can change that, and it provides specificity. And when the binding is perfect, when those sequences match up, the double-strand genome breaks. I'm just going to go back and start again with that one, just so you can catch it again. So we've got the CRISPR, the Cas9 molecule coming into the cell. It's unwinding the double-stranded genome. It's checking for a match between the guide RNA, the programmable element, and the genomic DNA. And then you see both strands of the DNA break. Now, at this point, almost every cell in the living world will say, whoa, 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 we have a break here. This molecule is no longer functional. It's broken. And the cell will try and repair it. And it will do it randomly. It will try and maintain the overall integrity of the genomic molecule by introducing a random piece of DNA. And this has the great function of sparing the function of the genomic molecule overall, but it disrupts that individual element. So if you target a specific gene, you can modify it. You can delete it. You can functionally knock it out. You can also insert DNA at the same time. So here we have what's called a donor molecule of DNA, something specifically designed to bind to that cut site and introduce a new functional element. And this is how you can conduct gain-of-function work, or you can overwrite existing genetic functions with others. So with this, you have a very, very precise tool, more or less, that allows you to ablate old function and introduce new function. This all happens in cells, in dishes, and it happens in the context of this genome editing protocol, where these are the steps that a scientist would take in order to execute this correctly. They would start by looking at the underlying sequence as a digital representation. They would look for possible guides where they can get that conformational match and precisely target an element and cut. And then they're trying to select for something which is computationally predicted to work very well at the on-target site, whilst avoiding what's called off-targets, places that you don't want to cut accidentally. You then synthesize these guide RNAs chemically. You can do this in a lab. And then you can complex them with something called a genome editing vector. It's basically a vehicle which allows you to introduce CRISPR elements into a cell with these custom elements, the guides, as well as the standard elements, the CRISPR molecules. They can be introduced in any number of ways. There's a lot of different vectors. You can use viruses. You can directly deliver them into cells. I won't go into the details. But ultimately, once those elements have been inserted into the cell reads them. It boots them up using this central dogma process of DNA to RNA to protein. You have those functional genome editing components in the cell. They do their job, and then you'll end up with a population of cells, some of which are edited correctly. Others are edited incorrectly because there are off-targets, and the vast majority of the cells will not actually be edited at all. So you'll have to select for the ones that you wanted. So we break this down into 3 broad categories of application of CRISPR or genome engineering in general. Cell line engineering is where the researcher is concerned primarily with modifying a cell in culture, and that's it. They do not want to generate whole organisms. They just want the cells edited at the other end. And cell line engineering, or model cell lines, is one of the mainstay activities here. So say you have a number of patients with a disease. They all show a characteristic mutation. The scientists would say, oh, I bet this mutation is involved in the disease. So what they can do is they can take a cell, introduce that specific mutation at the target site, and then see if these cells end up having the characteristics of those diseases. You have a model of the disease to conduct experiments on. In bioproduction, you're modifying a cell to produce chemicals or proteins in bulk, so you can purify those, sell them on for commercial value. And then in cell therapy, you're actually modifying the genes in order to exert a therapeutic effect at the end. And you can do that either by delivering edited cells to a patient or modifying their tissues directly. In whole organism engineering, very, very similar to what you see in cell engineering, the difference is you are aiming to get a whole organism that can reproduce with other edited organisms or other wild-type organisms, and they can propagate themselves over time. So you get multiple generations of edited organism. And again, we see the same kind of thing. Scientists are interested in doing this to have models of disease. You may edit a monkey to have a cancer, for instance. You may develop a commercial variant of an organism that can produce a chemical, a protein that you eat, or a protein for therapeutic in very, very high quantities. And there's a novel application which has really found a lot of interest in recent times because of CRISPR, which is the gene drive, the engineered gene drive. Here you're modifying an organism with the express goal of altering how it's going to pass on its DNA to other organisms. So typically, you take 2 organisms, they mate, they produce progeny, they'll have a certain distribution of how their genes are inherited. But with a gene drive, you can get what's called super Mendelian inheritance and drive into all of the offspring the precise trait that you were looking for. And this has great applications for pest control, which I'll get to in a moment. The final family of activity that we look at is screening. And in many ways, screening is similar to the engineering applications that I just walked you through. The difference is that with cell line and whole organism engineering, it's very deliberate. You know exactly what you want to do, what you want to edit. You do it in a small quantity, whereas with screening, you're randomly editing. You are editing using what's called a library, where you're designing potentially many millions of guide RNAs to modify potentially thousands of targets in a single organism. And then you look at how your edits cause a particular trait to be altered, and then you can work backwards and you can see how combinations of modifications could lead to certain characteristics. And from there, you can assign genotype to phenotype. You can understand how that organism works. You can do this in cells in a high throughput manner using computational tools, liquid handling, robotics, and you can do this in absolutely crazy scales. Or you could do this in organisms where you're editing a smaller group of organisms and you're really just limited on how many you can fit in your facility once they're adults, which is significantly less than the footprint. It's easier to have a lot more cells in a facility than to have a lot of adult animals, for instance. So in terms of how this breaks down into the industry, really, I guess there's two hemispheres. There's the biomedical research and the human health up top, where we're interested in understanding the function of genes and mutations, and then exploiting or applying that knowledge to medical interventions. And then the second hemisphere on the bottom is really about agriculture and industrial biotechnology, which is exploiting genome engineering in order to go more for these productive capabilities. And you can see the split is, by and large, oriented to human health and medicine. That's where the majority of the focus is on where we're getting useful applications from genome engineering. So with that, I'd say that's the essentials for understanding what CRISPR is and how it's being used. And now I'm going to hand over to Greg, who's going to walk you through the risks emanating from misuse of the technology.
Okay, great. As Eddie just highlighted, the flexibility, the precision, the versatility, ease of use, and the rapid diffusion of genome editing technologies has opened up a lot of opportunities for the peaceful, beneficial applications of this technology. But at the same time, it raises a number of concerns about how this technology can be misused. And in this report, we looked at misuse from a broad definition. We looked at the security, broadly defined, because we wanted to be able to capture areas where risks could have multiple different types of sources, and likewise look for policies that would be able to enable us to deal with multiple risks at the same time. So we looked at kind of four types of security concerns in general. One is biosafety, right? The effects of the accidental release of an edited organism into a population or into the environment. We looked at the role of dual-use research, which is research that generates information or knowledge or technology that could be, is designed to be used for peaceful, beneficial purposes, but could be misused or misapplied in ways that cause harm. We also looked at biosecurity, which is the deliberate misuse of the technology to cause harm in the context of either a state-run biological weapons program or the case of bioterrorism. And then lastly, looking at reckless applications, which is where the technology is being used in ways that are not fully thought through, consequences that are not anticipated, or people don't give due prudence and due consideration to the effects of their research or the effects of the products that they're selling. And so looking across these four different domains, these are the same types of concerns we've had for many years for other technologies. So we've had concerns going back to the 1970s with recombinant DNA in this respect. In the 2000s, the concerns were more about synthetic biology and some of the safety and security concerns revolving around that technology. But the report, we try and highlight how concerns about genome editing and CRISPR in particular are kind of quantitative and qualitatively different and create new challenges in this area. And one of the specific subsets of what we look at are the ways that genome editing technology enables different types of threats than what we are kind of traditionally used to. Now, there is the standard concern that these tools offer ways to modify microbes to be more dangerous. There's a long list of attributes that have been a long-standing concern that a nation state or a terrorist group could modify a pathogen to make it more lethal, more transmissible, some other attribute that make it more useful as a weapon. And this is a long-standing concern. This is what most of our biodefense programs writ large are designed to respond to. And CRISPR is one tool, but of many tools that have applicability in this space. Where CRISPR has a unique application or special advantages is in these other categories of risks that, as Eddie alluded to, deal more with affecting the source code directly, affecting the DNA of humans, plants, and animals directly, as opposed to enabling a pathogen that would cause harm if it infects the population. And so there are areas around what we call hijacking the microbiome, weaponizing gene therapy, and gene drives as weapons, where the same basic knowledge and processes that you would need to have in order to repair DNA that is causing disease or is causing a harmful condition, those same knowledge that you need for repair could also be used to disrupt normal functions of these genes and actually cause the disease of concern. So, for example, we now have a much greater understanding about how the human microbiome operates, how integral it is to many different biological systems and processes that we rely on. And that understanding, unfortunately, creates new vulnerabilities and new knowledge about what it would take to disrupt the homeostasis that we rely on for lots of functions. Likewise, in the area of gene therapy, understanding about how to manipulate and modify DNA to circumvent the immune system or to have effects in the nervous system or for other physiological processes, again, provides that insight and those capabilities in terms of the vectors that are needed to have a harmful effect if that's what's desired. And of course, with gene drives, while the emphasis obviously is on how do we either eradicate vectors that cause disease, how do we eradicate pests or invasive alien species, the ability to drive genes into a population that could be used to spread harmful genes in a population that would either have environmental consequences or would have direct human health impact if it's enabling a vector to spread disease more efficiently, spread new diseases, or live in areas where they're not currently viable. And what this collection of different types of risks really has in common is they challenge the traditional biodefense paradigm, which is very much focused on dealing with a small list of pathogens that we are concerned of being used as weapons. And so one of the major takeaways from this report is how genome editing is expanding the attack surface that we worry about from the context of biosecurity. The attack surface is a concept that comes out of the cybersecurity world, which is a way of kind of describing and measuring the number, the type, the accessibility, the consequences of vulnerabilities in code or in a computer program. And we're seeing something similar here where, given our much greater understanding and knowledge about how the code operates for humans and how that translates into RNA and then proteins, and our ability now with additional knowledge and additional tools that enable us to go in and edit that source code more easily, it creates more opportunities for that system to be hacked. And that hacking could be done for legitimate reasons, for all the applications that Eddie described, or it could be done by accident or for recklessly by people who are not taking into consideration the consequences of their actions, or again, it could be done in a deliberate way to cause harm to either humans, plants, or animals. And so this creates challenges for a system that has been designed to deal with a small list of pathogens in terms of medical countermeasurement development, detection, attribution. All of our dual-use research oversight systems are geared towards a list of pathogens. It does not cover much of the genetic editing research that we've been talking about so far. Just to add a caveat or qualifier here, the risks we look at, we're not saying this is going to happen tomorrow, although we've been surprised by things that are happening just the last week, so it's hard to rule things out. But there are still barriers to the use of the technology and to the misuse of this technology. There is still a large fundamental gap in our collective knowledge of how does genotype translate into phenotype. How do you actually take the knowledge of how genes operate and manipulate that in a way that will get a predictable result in the physical trait that you are looking for in an organism or in a cell? There are still, aside from the equipment and the materials that I talked about, there's still a good amount of tacit knowledge and skill required to use these tools successfully and effectively. And this is not just around the molecular biology techniques in a laboratory, but there's also a great deal of bioinformatics knowledge and skill that goes into designing the the genome editors and targeting them correctly. And then lastly, when we think about how do we go from an agent or an organism or gene editor in a test tube to it being something that is able to cause harm on a large scale out in the wild, there is a great amount of additional work and knowledge and materials that are needed for production, delivery, dissemination that go beyond just the narrow skill set that you need in a lab to actually build the gene editing vector that you need for your research or your application. So there are still barriers there. This is not something that we're talking about as being imminently feasible by unskilled amateurs, but it's also worth noting that all these barriers are falling. And in the report, we talk about some of the trends that we see in this field, in part because of this large demand for improvements to health, to agricultural productivity, for industrial uses. There's a huge amount of research that's being funded by governments, by universities, by the private sector. Its whole goal is to solve these barriers that were to use that we've described, but has that same dual-use application of also reducing the barriers to misuse. So again, this is a key area that's going to require kind of long-term attention to. Now, in order to kind of help illustrate some of these risks that we talk about in the report, and to try and put a little more, put these in more concrete terms, we developed some scenarios that are tied to these different domains of risks in terms of biosafety, dual-use research, reckless applications, and biosecurity concerns. And to talk about some of those scenarios, I will turn this over to Megan.
Thanks. So as Greg mentioned, one of the things we wanted to do was to develop tools to really begin to think through the complexities of the governance challenges that exist in this space and strategies that we might employ in order to counter them. And as David also mentioned, we thought about this project really not just as a product, but as a process. So the co-development of these scenarios was one of the tools we used in order to begin to imagine the types of things that might be on the horizon. So these are each borrowing from a number of these different risk areas, from dual use, from biosafety, biosecurity, and reckless actors. But there's really a lot of sort of crossover between the actors and the aims. There was a number of criteria we used to develop the scenarios and to select them. Primarily, they were used to illustrate some of the less intuitive governance gaps, but also to highlight the diversity of actors and motivations, technical capabilities that might be employed, particular technology applications of interest. We also wanted specifically to look at things that were sort of plausible but not immediately possible, because there was a lot of concern in this study, as there are in others, around the potential information hazards that might be coupled to the development of scenarios. And lastly, they really weren't worst-case scenarios in terms of the potential number of individual sort of casualties that might be employed, but really to begin to look at these sort of less intuitive gaps. So these are just some illustrations. There was 6 scenarios that are developed in the report. There's others that could be imagined here, but they are a place to start. I'm going to walk through just 3 of them in a little bit more detail and highlight the others. So the first of these we call CRISPR charlatans, and it is even more relevant this week, perhaps, than a couple weeks ago. This is a scenario where we imagined there might be a small sort of outbreak of inflammatory bowel disease-like symptoms that occur locally. And then over time, it seems to spread quite quickly across the country. And as we began to unravel the source of these conditions, it becomes evident that all of these individuals have been taking the same probiotic. Going one step further, that probiotic, it finds out that the firm actually, inspired by the claims of what might be possible by modifying the bacteria in that probiotic, decided to contract out to a research organization that puts in an immune modulator into the probiotic. And this is what is causing some of these conditions. One of the reasons that we wanted to highlight this is to show the diversity of new types of products that might be imagined using genome editing in order to enable their development. And these are exposing really the attack surface that is our bodies in new ways of delivery and dissemination that might be taking advantage of areas that are more loosely regulated. In this example, a probiotic that sort of falls between a food and a drug, especially if it's not making medical claims. Another thing that we wanted to highlight is the ability to actually contract out to research organizations that might do these types of things on demand and with less oversight. As we begin to look at the types of policy options, and these are just examples, there's ones in the book, it shows the increasing importance as these tools are developed and used in a number of different sectors to engage industry in co-developing the types of oversight that might be required, especially taking advantage of actors that really do want to safeguard their industry to figure out where those gaps are, and particular high-risk consumer goods. We talked about delivery as being one of the key bottlenecks. Some of those delivery and computational design elements of the process might be ripe for more oversight. But of course, coupled to this is just overall strains in regulatory capacity. So how do we enable different regulatory agencies to work together to identify these risks? One of the second scenarios we developed, we called weaponized bio-narratives. And this scenario revolves around a malaria lab that's looking into one of these technologies, gene drives, to develop a self-propagating system that might be able to either sterilize the vector or cause it to not be able to propagate the parasite. And in this case, we look at an actor, an environmental group that happens to know one of the lab members, steals their card, and does a small-scale release of these agents. It's actually in a place where it's winter. It's not likely to propagate. But what does propagate is the story. So it's videotaped that begins to be picked up by different groups, knowing that this is already a polarizing narrative. It's a story that equally propagates by even state-sponsored misinformation campaigns. So even though there isn't a physical effect, the psychological and ultimately the effect on the economy and on science is quite large, because there's a distrust of the technology that could have sort of widespread beneficial applications. And so here the story was really around, we're dealing more and more with not just risky technologies, but risky communications. The uncertainty that David spoke about can itself really be used to polarize and seed fear and doubt, even without a physical effect. And secondly, that there are many different actors with many different motivations that may be leveraged in order to make an event, and really a political event. And the third that I'll give some detail around was around the development of new types of biological weapons that have new strategic uses. And in this case, a dissident that is targeted, and the regime that's going after them is interested in a way that's more covert to assassinate that individual. And so they take advantage of published research to look at an agent that might be able to cause a neurodegenerative disease that might be less easily detectable. And that initial test that's successful might inspire further programs. And so here, again, it's our initial calculus around traditional biological weapons programs may no longer hold with some of the new types of capabilities we're beginning to see with these technologies. So we have to begin to reevaluate the initial assumptions behind this. And also the idea that the footprint, the scale of these types of programs may differ widely because of the availability of a wide amount of public research that has increasing dual-use potential. I won't go through the other ones in detail, but some of the other, and I'll just say we're on the edge of the Biological Weapons Convention meeting of states parties this week. And so this is one of the topics, genome editing, genetic weapons, and gene drives that is on the docket. And it increasingly begins to bring our focus to the need to do landscaping, science and technology review, understand how these new types of capabilities and new types of actors affect our agreements around what we will do or not do. So some of the other scenarios, we're increasing around vulnerabilities in the agriculture and commercial sector, developing new types of disease-resistant organisms like pigs that might be reservoirs for human disease. The idea of new types of bioterrorism, being able to contract out to commercial firms to develop pathogenic bacteria out of non-pathogenic bacteria, using these new types of tools, but perhaps familiar ways to deliver them. And biosafety incidents that can occur from labs that are developing these technologies and all of the infrastructure around them that may not recognize immediately the types of risks inherent at the forefront of knowledge, just because they're unfamiliar and there's really a dearth of expertise on knowing how to handle these things in the midst of uncertainty. So collectively, these scenarios are really meant to illustrate, the number of different types of risks, the number of actors that are involved in both posing and in mitigating those risks, and the types of strategies that we might employ and to begin to imagine the things that are on the horizon. Now I'm going to just hand it over to Jesse to wrap us up.
Great. So the question is, what are some of the key takeaways from this study? And we really can group them into two categories. The first is those that are specific, again, to genome editing in particular. That's genome editing as a focus. And then the second is those that speak to some broader trends in the life sciences and biosecurity in particular. So if we see here, we've discussed many of the benefits that both Eddie and Greg have touched upon. And we wanted to make clear at the outset that it is extremely important to recognize that genome editing could play a critical role in improving the human condition, whether it's through increasing crop hardiness, greater agricultural yields, breakthroughs in advances for medicine, improvements to the economy, and so on and so forth. So our goal was at the outset to make explicitly clear that there are benefits, but we also have to safeguard the risks. The second is that it's clear that CRISPR and genome editing, that this technology has and will continue to disrupt the biosecurity landscape. One way, as Greg mentioned and Megan mentioned and others, is that we found in the report is that the attack surface is going to be increasing. Whether it's through different paths of exploitation, different actors in this space, or ways that we possibly couldn't imagine, this landscape is shifting and we need to be aware of that and safeguard against it. The third is that the study also suggests to us that there are broader trends in the landscape that we need to pay attention to. Now, the only way that we're going to be aware of these broader trends is if we take the technology seriously. So we endeavored in this study to really do a deep dive. I mean, that's why many of you who are not trained in the life sciences, sat through a good bit of this review of this study that is technically specific. The goal here is that really the devil is in the details. That in order for us to be able to think about both genome editing and the broader picture, is that we have to focus on the specifics as well. Only through sort of looking at the small part of topography can we zoom out and get a better sense of what this landscape holds. And in doing so, we recognize, and this was one of the attempts in illustrating this through the scenarios and through the sort of the different actors that we highlighted and that we explored in the study, is that there are going to be different stakeholders than we've seen in the past. Part of this is simply, or a large part of this is simply just a function of this technology, what it makes capable, and what it enables. And that's in part the composition of this research team, is that this is both by function and design that we have policy experts, security experts, medical doctor, technology expert, and ethicist. The idea is that we need to be able to to engage these key stakeholders in various areas and be able to speak to them in their own language. Last, is this last? No, last but not least, is that in terms of thinking about the governance options, and Megan mentioned just a few, just got a taste. There are many more in the full report. But that our call is that more applied research is needed. And being able to prototype and test different types of governance measures before the stakes are too high. And so be able to think about how to incentivize actors to act well, both through push and pull factors, being able to engage social scientists, those in the tech community, and so on and so forth, to adopt some of these, structure them, think about them, and move forward in productively designing types of policy options that can be widely adopted and accepted. And so in thinking about that, we really crafted a kind of a two-prong approach to a path forward. And this isn't just one path, there are many. Right? That there is, again, and the governance options that we present are not the end-all of be-all, they're options, and there are many. But the first is that we need to focus on a strategy of collaboration. And again, it's bringing people like this, people like the collection in this room to bear on these tough problems, right, to engage stakeholders. So that's members of the government, policy wonks, security experts, and those that are working in the science. So we need to go from bench side to bioethics. And the second is that while working and endeavoring to have that collaborative, engaged approach, we also need to hedge against the possibility that there may be failures and gaps, and they're likely to occur. And so what might we want to do about that? Well, one option is that we need to use technology, and we need to use innovation as a security strategy. So we need to harness the technology, such as genome editing and future technologies, to safeguard against the very risks that they may present. So I'll just give one or two examples of how this could occur. There are a few government programs that are investing heavily in designing technologies using genome editing that can detect uses, harmful, deleterious uses of the technology, that can remediate, try to bring the environment back to a sort of genetic baseline. And these are defensive technologies. So one path forward is to think about how we can bootstrap the use of this technology, because it's not going away, into creating and crafting better biosecurity strategy and policy. So a few of the acknowledgments here. We have our project contributors, had a host of research assistants. One co-author who is in this room but is not on stage I'd like to thank in particular, and that's Sarah Denton. And we had many research assistants, institutions, and workshop participants. A real quick plug, all of the issue briefs are available online at editingbiosecurity.org, as are the working papers. And if you are unable or if you want to save the paper, the full reports are also available as well. I think with that, we will conclude and take questions.
Sure. So we'll open to questions from all of you. Briefly, if you can state your name and your affiliation. And if you want to direct a question to a particular person, please do that. And so we'll just open the floor. And there's a mic that's traveling around, so just raise your hand.
Everyone who shuts up. My name is George Robertson, and I'm an advisor to the Joint Program Executive Office, Chemical and Biological Defense of the U.S. government. One of the things when I just briefly looked through your report is risk needs to be quantified. And I believe there are two risks, one of which is what I'll call the risk of the project, which is what you quite well described here today. And the other is the risk of management, the risk of oversight. And in an earlier life, I was working with overseas vaccine developing manufacturers. And the emphasis on quality was top down. If the manager wanted it, you had good quality. If they didn't, they didn't. And part of my job at that point was to change that attitude. But I'd like to query the panel on, frankly, if you can, which is more important? The risk of the project or the risk of the oversight and perhaps the deliberate or accidental release issue or those sort of things. So thank you. That wasn't planned.
I'll take a stab at that. I don't think it's necessarily a matter of one or the other. I think there are different mechanisms that you'd want to have in place to mitigate risks posed by a specific project, and then programs in place to ensure that oversight is done consistently and with high standards, and that you don't have the risks that come out of lack of oversight. So you could have a more generic process that is a matter of kind of educating the scientists who are engaged in these types of projects so they understand the nature of the risks that are potentially manifesting themselves. And so that would require the people in the trenches, in the labs, at the bench to be very cognizant of this. But at the same time, there does need to be some top-down in terms of, okay, here's the guidance, here are the standards, here's what's expected of you. And again, that's part of the collaborative approach that we would hope can flow out of this, is you have both people from the science technology side and from the policy side coming up with what are the appropriate types of oversight that would raise the awareness, would mitigate some of those risks, but would not be so burdensome as to get in the way of the kind of research that is very, very valuable. But those, finding that balance is not easy. It's not automatic. It's going to require a good deal of dialogue among these different stakeholders to get there. And one of the key takeaways from our report is that we need to start that conversation now. We don't want to wait till after there is a biosafety incident. We don't wait till after there is some major well, there already has been another major regulatory failure or oversight failure or biosecurity incident. That it's much better to have those conversations now when we can kind of plan ahead and not get caught in the reactive trap that happens after a major event.
I'll just follow on by saying I think that it's a core challenge of how do you ensure that the leaders of the increasingly diverse number of organizations that are conducting this work can prioritize security as a topic. It's something that in the life sciences establishment is not always top of mind and isn't necessarily natural. And so part of these projects even was to give individuals who are working across a diversity of organizations the chance to work on something proactively where they become more of a leader in thinking through the gaps instead of a real sort of compliance mindset that leads to other types of problems. And so that's why part of the project was really focused on this process of what do you need to actually equip people to feel like they can take ownership of whether it's a project or a process management issue.
George, I would also add, yeah, of course we do not want to see this viewed as an either-or proposition. I'm also mindful of the realities which often result in that kind of trade-off happening. It's sometimes a function of the kind of mechanism that's deployed for the oversight, meaning law, for example, hard to revoke or change. Guidelines, more easy to do so. And so sometimes the choice of mechanism might then allow for less of a either-or and more of a flexible, iterative, adaptive kind of approach where both can work together well. But it's a very important point, and you only have to go out to NIH and talk to folks who have tried some experiments in registration, for example, and have data that show that even the most mild form of registration for use of a database results in clear evidence of diminished use and lack of beneficial work. So it's a really important point.
Yeah, I mean, A lot of the work that's, thank you. A lot of the work that's been described, of course, has been done by other means, lentiviruses, plasmids, et cetera. However, this is far more, I think, exciting for what can be done, because many of those insertions into the genome are random. And as we all know, they can have some bad effects, unintended effects. So certainly in the US, EU, there are institutional biosafety committees. But as you and I were talking beforehand, David, there's no real Australia group for how we can track this, because now it's commodity items at Biorad or something like that. So but the idea of understanding and the depth of it clearly, especially to the biosafety community to start, I believe is extraordinarily important. Thank you.
Thank you. I'm a reporter, Sun Hao Liu, a reporter from Hong Kong Phoenix TV. I have a question for Dr. Relman and also other experts. As we all know, there is a group of Chinese experts, Chinese scientists, created 2 babies by gene editing. What's your comments on that? Do you think that's a breakthrough, or do you have worries in this case? And also, do you think there's a universal standard for gene editing in the world? Thank you.
Well, as you know, the events of this past week have engendered a lot of discussion. I'm happy to make a few comments speaking as an individual, but perhaps in the spirit of the report as well. As an individual, I'm greatly concerned that there doesn't appear to have been the kind of open, pre-planned discussion of what might have been the motivations, the options, and then the approach to addressing what might be some real questions and potential beneficial purposes. Without that kind of discussion, you end up with a lot of ad hoc activity, which is sometimes hard to reverse. Once you pass certain lines, you don't walk back. And I think some lines were crossed here, not of a technical sort, but of an ethical. And is this what we as a larger set of interconnected societies want for ourselves? That was transgressed. That was violated in an ad hoc way, without the kind of discussion that this report seeks to clarify and describe, because there are many people with viewpoints that were not adequately reflected and represented when early decisions were made to do these experiments, at least judging from what's been made publicly available. A lot of people caught by surprise. And I think when you look at the kinds of any of these complicated scenarios and think about what are all the kinds of viewpoints and vested interests that deserve a place in a conversation, many would seem to have been left out of this.
Hi there. My name is Mei Fong, and I write a lot about China, not so much about science. And I'm kind of curious, this whole discussion about the CRISPR technology has sort of illustrated, the big debate between what can be done and what should be done, clearly. But my question pertains more to who, which is, you've clearly all outlined a very big need for oversight across industries, but clearly as what's happened with China, there's also a need for oversight across borders. And so my question would be, have you identified any potential bodies that should be having oversight over this? Because clearly we say there should be, but who's going to be doing it? And how can that be done? And I know this is a bit of a difficult question, bearing in mind this intense global rivalry with every country vying to gain an edge in biotech.
Let me make a comment. So I think that increasingly, science and technology is a really global enterprise, and so the communities who are involved right, at least at the stage of research and development, are often sharing a lot of this information. And so I think what we're going to see is not just one central body that's going to be helping to figure out what the should we do or develop norms, but it's going to be a number of bodies that are trying to set strong norms. But increasingly, I think we're dealing with a sort of network governance regime where it's who are the communities, right, that individuals working in this space identify with, and where do they take their cues? So a few people in this room, myself included, have worked with organizations like the International Genetically Engineered Machine Competition that this year involves, I think, 340 teams from 48 countries who are involved. And actually, almost a third of those teams were from China. These are teams that all are coming together to share their discoveries, right, about what they're doing, share the cutting edge of what's happening. It's also been coupled to programs like the one that Ed Yu here helped develop with the FBI, sponsoring some of these programs so that when all of these individuals who are operating see something they think is not quite right, it's not just awareness building. They have a whole community of first peers that they can speak to about what might be right, and also are plugged into professionals and networks whose responsibilities are around safeguarding individual countries and individual organizations. And so I think that increasingly actually having eyes on what's happening in this space is going to be just as important as, say, an oversight capacity somewhere. But we can't get away from the fact that this is a technology that everybody wants to develop because of its promising benefits.
There is no particular strong governing bodies with oversight over this process right now, or appears to will be in the future that we see?
There are several strong governing oversight bodies. You can maybe pitch in, but right, just this week with the Biological Weapons Convention, talk about a strong norm, right, around a technology that we are not using for offensive purposes. Right? And that's very different than other areas of technology. And so those types of institutions really are the foundation on which all of these other types of capacities and actions are built.
Yeah, I mean, one of the more interesting findings from the report is just how many diverse applications are of the technology. And clearly, the use of genome editing for germline editing has kind of become front and center because of what happened in China recently. But there's a myriad of other applications. And we do have some pretty strong norms and red lines on some of them. So the idea of using genetically modified organism as a weapon, right? There was a red line that the Biological Weapons Convention says is prohibited, right? Most countries in the world have signed up to this, so this is not something that's kind of up for debate or that requires any kind of consensus building. So there are some very strong norms out there. There have been attempts to build some norms around things like human germline editing, which have not gotten institutionalized, but there have been different groups that have kind of promoted the idea that we need a moratorium or we need to have certain criteria in place or conditions met before this is an acceptable type of research to go forward with. But I think that is still in an earlier phase. What I would say is that the response we've seen so far from authorities in China and from relevant authorities in the US has been encouraging in the sense that no one has kind of dismissed this or downplayed it or marginalized it. They've said this is an important issue. We need to investigate, and we need to understand what happened, why it happened, and is there something wrong with our processes, or do we need stronger norms in this case? So I think now we're asking these questions in a way that hopefully will be productive and fruitful. And I think one of the other kind of takeaways is that this kind of conversation we need to have in a couple of these different areas as well, right? Not just wait for someone to do something that transgresses one of these boundaries, but instead we need to be having these conversations ahead of time and trying to establish some of these norms and red lines and limits early on and make it clearly communicate to the scientists what's kind of expected, and for the policymakers to do this in a way that is productive and is not getting in the way of legitimate beneficial research. And again, that's why we need to have this collaborative approach both between disciplines, between countries, in order to have that be something that is an enduring way of managing the different ethical and social risks that this technology poses?
I think I want to come back to what you said earlier, David, which is that it's key to remember that nations have a plurality of views from the different publics that reside inside of them. And I used to think, back when CRISPR arrived on the block, damn, we need an international treaty where we can just deal with this up front and avoid any issues happening. The trouble I realized with that is that, one, that would take quite a long time for nations to negotiate effectively. And second, how can nations accurately reflect the opinions of their citizens on this technology? And this is complicated when the technology has so many different applications in different spaces, right? The views on gene drives in nation A could be completely different from gene drives in nation B, and they may have divergent views on germline editing or clinical uses. In terms of an effective strategy I've seen in the UK for understanding how publics respond to technologies and how they demand it be used, I would point to the example of the British Human Fertilization and Embryology Authority, which has conducted some very in-depth studies on attitudes to genetic engineering of humans with regard to mitochondrial donation therapy, where 3 parents provide genetic material in order to produce a single offspring. And the UK, I believe, spent about 10 years running several workshops with religious leaders, patients, average citizens, scientists, parliamentarians, in order to get a very deep understanding of what the national view was in order for Parliament and others to make an effective decision at the end of it. Now, when it comes to what we saw in China last week, I'm still on the fence. And the reason for that is because I think when you think about genome engineering on a long enough timeline, I would say, sure, we'll probably end up using it in humans. The question is, how long will it take for us to get there effectively, where we're all bringing each other along for the ride and we're all comfortable with it? And I would say that if we do move towards a treaty or something a few rungs down, it might make the most sense to outlaw things quite brutally initially, but also give us the flexibility to reinstate applications as they become more acceptable in a society as the years and decades roll on. And I think you will see the change in attitudes over the coming decades. It may even be less.
Thank you very much. Terry Taylor from the International Council for the Life Sciences. Thank you very much for the report, which I look forward to reading in detail, and your comments. My question comes from a policy implementation point of view rather than a scientist point of view. And I was, it's about the international regulation rather than domestic regulation. And I wonder what you would see, any of you would see, as the first 1 or 2 steps to improve the international regulatory environment. I'm a little bit of a skeptic because of my personal experience in this area. I was a little bit uneasy when I saw in your report you said about looking at the scope of the Biological Weapons Convention, its coverage, I think the word is. That makes me uneasy as a former negotiator, that more definition results in a narrowing down. That would be my big fear. It's a political risk would be involved in doing that. But, and really listening to the last remarks, sir, by you, should we be looking at an evolutionary process? So there's a whole bottom-up process. Maybe it's a matter of timing. Maybe the time is not right to have an international convention. Should there be something new? What would it be? And we need to look at the other existing instruments that already exist, like the Convention on Biodiversity, the Resolution 1540 of the UN Security Council, these other things. So we have to look across the landscape. Maybe there's a time for another Asilomar conference of that kind, galvanize internationally the scientific community, which that had a big impact, of course, at its time. So I wonder about your views on that. Thank you very much.
I might just make one comment. I think what we've seen is within what is often a usefully flexibly large umbrella of these international agreements, a lot of the difficulty does come at the sort of level of implementation and how to carry forward what are often sort of broad recommendations or broad coverage and scope. And so I've been personally heartened to see a lot of the partnering initiatives that have brought together 2 or more countries, right, to share their efforts on how to, say, develop a dual-use research of concern oversight, something that has sort of plagued us for a long time, to share, how did they define it where they were, how did they reach out to different groups, how can you use these same types of tools. And so I think there needs to be a lot more sharing of resources and approaches at that sort of ground level of what might be possible. And I think there's only so much you can do always at the sort of highest level, and there needs to be a lot more sort of resources and attention put into partnering and information sharing. But that's a personal opinion.
Yeah, I would add, so you mentioned the CBD, the Convention on Biological Diversity. So Megan was talking about sort of bottom up, so I'll speak to the top just a little bit. As you likely know, there was just a meeting in which gene drive was under discussion. There was the threat of a moratorium on the research, and that did not occur. So the research is able to go on under sort of narrow circumstances. So that's one example in which it's in which there is a international treaty body that is taking up these topics. The other was the BWC, and you had a point about the narrowing of the scope, and I think that's a legitimate concern. But there are certainly, I take your point to be, or part of your point to be, in which I agree, is that, we might do well to look at existing treaty bodies, existing institutions that in which this might fall under the purview or scope of oversight, right? As opposed to, say, recreating the wheel. So I would say it's one potential risk with starting at the top is that you may not get the stakeholders like Megan mentions, who often are the ones that are in the trenches and are going to be needed to do this research, to implement it and follow the guidelines and rules.
Also, if I could just address the specific point about the BWC, definitions are again, devil in detail kind of matter, but more fundamental is a need for that community of diplomats and stakeholders to even know what is happening in the realm of genome editing. And so this idea that's been floating around for a long time that is not yet to be implemented has been establishing a mechanism within or connected to the BWC that does regular systematic reviews of science and technology relevant to the convention. And as we talked about, CRISPR and genome editing are just one of many emerging technologies that are going to have an impact in this space, looking at automation, synthetic biology, artificial intelligence. And so having a way of looking across those technologies for the purposes of identifying where are potential risks and to what extent do we need to not change the nature of the treaty, but have an agreement among the state parties that the kind of general purpose criteria covers all the things that we're worried about. That's a valuable function. And so I think there are a lot of things that are existing institutions, BWC, Convention on Biodiversity, that need to be updated in a way to take into account these new technologies that no one even thought of when these were invented. And not that it can't be done, but it's just we need to put some effort into doing that. At the same time, we don't necessarily need an international convention with 193 state parties in order to deal with some of these issues. So when we're looking at human germline editing, for example, it might be easier to instead of having a UN-based treaty, if you have who are the 4 or 5 countries that are the biggest funders of life science research, right? US, EU, Japan, China. If they're able to agree amongst themselves to certain standards or conditions or criteria that are needed before countries are willing to sign off on human germline editing or the use of gene drives, that would go a long way towards creating some kind of standard that eventually could be broad and other countries could buy into. So I think there are a couple different ways at the international level. It doesn't have to be universal to start with. There can be multinational approaches as well, but with the goal of making it universal over time, but you maybe need to start with a smaller effort. And again, going back to a point that Megan made earlier, this is part of the reason why there's a need for this kind of iterative approach to policy building in this regard, right? Having this kind of design, build, test approach to governance in here is important to do things at a small scale or limited scale to see what works and what doesn't work, and and looking at natural experiments like what has happened in the UK for other countries to learn from that experience and be able to replicate the things that work and avoid mistakes that are made with either overregulation or gaps or bad definitions. So again, this is going to be an ongoing process with multiple paths getting to that same destination. That's kind of the roadmap is what we've been trying to provide, not the exact exit you're getting off at on the journey.
I have Fivu with the Biotechnology Innovation Organization. Thank you for everyone's time and your work in this important area. I also have two questions. The first one, there was a mention of key stakeholder engagement and specifically industry in a regulatory context. I was just wondering very briefly what level of engagement or representation from industry was available in terms of providing perspectives and insight in terms of informing the findings of the report and all the white papers that were on the screen. I haven't had a chance to review the reports myself. And then the second question is more broad, and so I don't think anybody here would disagree that gene editing technology and CRISPR, specifically has enormous potential to benefit the human condition. So, the question is, how do you balance, the need to innovate? Because we talk about applying this research, but gene drives and some of these other applications are just scratching the surface, right? And so, how do you balance the need for innovation with addressing some of the risks? And just as an anecdote, we spend a lot of time on the Hill and talking to policymakers, and what we're hearing from staffers is like all we're hearing are negative things about gene editing. Like we only hear the negative things, and in the news it takes something that happened earlier this week for gene editing to really hit the news, and they're not really understanding the promise and the benefits of this technology. And so that's where we come in. We help educate a little bit in terms of the science and the application, and so just broadly, just kind of your comments on how we keep that in mind as we move forward.
Do you guys want me to take it? Just a couple comments. Please chime in. So I mean, this study itself was sort of fairly limited in terms of total number of people who are involved, but we were lucky to have a few individuals. One that was from one of the sort of leading synthetic biology firms, Ginkgo Bioworks, who helped author or comment on one of the working papers. Eddie himself was part of a firm that provided some of the services and tools that are required in order to do these types of projects. Then we had also individuals who are working in labs but have been involved, and those who have been consultants to firms to deal with many of their policy and regulatory issues, even just within that small group. But coupled to that actually has been the importance of having a few individuals who have gone through some of these sort of security awareness building programs who then are still within the firms so that when they get a call to participate in something like a security thing, they're not scared off, but they think, oh, how does this let me protect my business and actually maybe even build new business? So that's been actually a big part of the second step of things, is having a lot of these firms now work with new types of government programs, like the ones that have been piloted through DARPA and IARPA and others, to actually see how they can build platforms for manufacturing countermeasures or can contribute to the research behind that. So that's been a big part of at least sort of building business around solving some of these security problems. But I'll agree that stories like what came out this week, unfortunately, they do have an impact on the industry as a whole when it sheds a negative light on what is really a pretty amazing, beneficial new trajectory in the life sciences. So I don't know. Maybe we need to work together more.
And also just to acknowledge Sarah Carter, who's in the audience, wrote our issue brief on the bioeconomy and CRISPR and biosecurity. So we tried to draw upon the expertise of people, in multiple ways. So we're, as you'll see in the final report, cites her work quite a bit. So thank you, Sarah. On the issue of balancing risks and benefits, this is what we call the dual-use dilemma, right? It's a dilemma. There is no solution. If there was, we'd be there already, right? We've been struggling with this issue of balancing risks and benefits in life sciences, actively working on this for the last 18 years, with various iterations of National Academy of Science reports, U.S. government policy, and it is constantly a work in progress. And unfortunately, what we keep finding is, as the technology evolves, as there's a new experiment, as there's a new event, we realize that the framework we have is not sufficient. And so one of our working papers goes through this, looking at the kind of the history of risk assessment in this regard, and looking at how we've tried to balance these risks and benefits. It also looks at the governance, the history of governance in dual-use biotechnologies, and how we've tried to struggle to adapt governance in order to deal with these emerging threats. And not that we never get it right, but it's a constant struggle. And we're now in kind of the early phase of dealing with that very thorny problem in regards to genome editing. And what I'd say is two things. A, right, you need institutions that do this for a living. Right, you need to have this be part of a constant, dedicated organization that's doing this at the international level, related to BWC, or within the U.S. government that is focused on understanding the nature of these technologies and what impacts they have on safety, the economy, health, and what have you. And again, that's something that we're lacking. So it winds up being very ad hoc because it's really up to the individual stakeholders to do this without having this larger institutional framework. And there are a couple of policy options described in the report that will give you a couple insights into particular options we could use to expand oversight.
I would just add, I mean, this isn't about balancing it necessarily, but in the report, I mean, we provide a number of representative concrete actions that could be used to mitigate some of the risks in order to preserve the benefits, right? So an example would be best practices for biosafety, right? With gene drive research, for example, right? It's not required. The level of awareness among some actors may be relatively low, given the sort of the somewhat nascent nature of the technology. But sort of updating and engaging in best practices. That could be one, because it's going to affect your bottom line, not you necessarily, but the global you bottom line of those in the industry if there's an accidental release or a biosafety lapse. Another is that Megan talked about the bio-narrative, the information warfare scenario, which you have what is, a relatively innocuous kind of a release that results in, people who might otherwise have small voices get the megaphone of social media and can really start to sow fear. So one of the recommendations that we make is that social media platforms and the fact-checking organizations in which they rely upon that they have those that are expert in life sciences, which they don't currently. Right? And if they did, I know it's a bigger problem, right, with the meddling in elections and so on and so forth, but even that. So these aren't simple fixes, but they're concrete options that could be used to not necessarily do the balancing, but reduce some of the blind spots so that the benefits don't take such a hit from things that, from occurrences that could otherwise be mitigated or avoided.
So I'd like to ask a question that's somewhat related, which is, there are all sorts of, some aspects of the regulation and the like, and guidelines are technical and directed towards technical audiences of scientists and the like. But throughout this discussion and in your report, is this question of storytelling about this technology or narratives about this technology. It was great that you picked an example that brings some of that up. But I'm just curious, if you can, to reflect on what you think are the more appropriate narrative directions for a broader public to make sense of these technologies. On the one hand, there's a lot of logic to the dangerous narratives, right? They might provide one of the more powerful calming effects. Certainly that's true for chemical and biological weapons. Mm-hmm. Those narratives are crisp in our minds and have helped motivate and enable the consistency of that regulatory process, right, for decades. But there might be a sense of concern about negative narratives. So what do you think ought to be the direction of public narratives about CRISPR technology moving forward?
I think there's sometimes a human tendency to want to place value or make a value judgment on a technology when in fact, or try to control it, when in fact it's really more about the intentions and purposes of those who seek to use them for whatever the range of possible outcomes and effects. And so to me, when we talk about applied research, as a biologist, I'm almost inclined to say where we really need much greater understanding and study is in behavioral science. What is it that motivates those who would now be in a position to use these technologies? What are the incentives that might change the way in which they make decisions when they get up in the morning and decide to do this experiment or that, or invest in this particular product or that one? And when we understand that better, I think we will have a much greater ability to help guide the beneficial uses and use terms that are more productive in helping people understand what is it that we're all trying to do together, rather than slapping a negative label on something which simply can't be labeled as such. An organization like yours, I think, to get back to your question, is a really important one for helping to propagate the kind of discussion and focus and incentive awareness that would be useful.
I think, unfortunately, we see this through pretty much all of the stories that are told about this field. It's the same story in the 1970s when genetic engineering, first generation genetic engineering, it was promise or peril. And it's this very same one today. My colleague calls this the sort of half pipe of doom, that bioengineers promise to save ourselves from the world that we're destroying, right? And often, I think the story that's more appropriate is to say, actually, we're already hugely reliant upon biotechnologies in our economy and for our livelihoods. And the idea, it's often the boring applications in materials production, in the types of fuels we might develop in our healthcare already. And the idea that sort of biotechnology is here already, and it's something that we need to sort of protect and usher forward. And often boring is kind of best, right? It's the boring but essential applications that are the ones that perhaps are going to make our lives better and save ourselves. And so writing that story around boring is best. I need to find a good news writer that will be able to make boring exciting. But I think that idea that it's already here, it's not something that's already on the horizon.
I agree with that, with all the percents that I can fathom. I think there's such a tendency to sensationalize emerging biotechnology, and I think that a very positive change in the narrative would be for scientists to no longer talk about how CRISPR is easy, because when that narrative is out there, it leads to reactive assessments that get the technology wrong, that assume that any person can pick up a pipette and start engineering their own genomes, which leads to reactive media, and that will filter into policymakers, which mean that the trajectory of the technology will ultimately be altered because of these concepts. Rather, we should be very specific about which aspects of the technology are easier to use than other aspects of the same technology or older generations of the technology. And I think we unfortunately find ourselves in a position now where this CRISPR craze, the hype, suggests that it will change everything in just a few short years. And really, we will see big changes, of course, but they are going to take a much longer time to materialize across all of society than is widely suggested at this time.
I think maybe the information technologies analogy is good. It has enabled a lot of things, right, to connect us, see what we can do with it. It's a platform that enables many things. But when it spreads, it has some unintended effects that we have a difficult time controlling.
I would say that, it's hard to write a good story. So that's was one of the real challenges of writing this report is that it doesn't make for good pull quotes to do kind of boring is best in terms of it's hard to tell a good story when if you want to talk about what is faster, cheaper, then you have to qualify it and explain how, well, it's faster compared to X, Y, and Z and all of these conditionals. But I think what makes a good story is when the tech is right and it's real and it's described in a way that is approachable but plausible. One of the stories that we assiduously avoided was that of the super soldier, right? Just breeding generation upon generation of soldiers and pushing genes through them. It's a great story for science fiction, but it's a bad story to kind of spotlight the issues and concerns that we want to be thinking about. So I think that we didn't get it perfect, but the criteria that we laid out for storytelling, I think, was to me satisfying. I mean, there were some areas that were wanting, but yeah. So I would say that it needs to be probable. The actors involved need to also be real. And I would say that the types of stories we want to tell are stories across a wide range of spectrum that indicate potential risks and concerns and anxieties, but then also the benefits as well.
Hi, good afternoon. Ed Yu from the FBI Weapons of Mass Destruction Directorate. More of a comment than anything else. I just want to applaud you all for the completion of this project, and if anything, you helped me come away by coining a phrase today. It's mind the policy gap. Because at the end of the day, CRISPR-Cas9 is just a tool. This is probably going to get me into hot water, but it is just a tool. And I think what you all were able to highlight is that this is so much more than just looking at pathogens and what I would say the traditional debate about dual-use research concern, that there are going to be other applications and it's going to be very much contextual beyond just the health aspect of it. And so there's going to be the commercial, there's going to be the foreign policy, the foreign diplomacy, the foreign trade elements. And if anything, the real highlight for me is that the group of experts that you brought together would be a fantastic IBC of the future. I mean, that's what the makeup is going to have to look like. You're going to have ethicists, international policy experts, as well as the scientists and the security experts all combined, because that's where biotechnology is taking us. And if anything, we're talking about a designer baby event that launched this new round of conversations, not the modification of a pathogen that causes a pandemic. So I think that's really something that should really hit home to a lot of people when it comes to the biosecurity discussion, that this is a different take. And applaud you all for convening a really good and important diverse group.
Thanks.
Yeah, my name is Khalil. I report for different viewers, the Middle East Broadcasting Network. So this topic is kind of new for them. So it's just, does this debate look like the same one as the one before the starting of using the nuclear or the atomic energy, and then they created the agency, the International Atomic Agency, under the umbrella of the United Nations? And now, and the second question is about what's the current mechanism to deal with such, in case, let's say, something happened in Taiwan or in Vietnam or in South Africa, somebody used this technology, the CRISPR, for a political use inside that country. So right now, is there any system that make the international community dealing fast and in a way that they can stop this kind of misuse, efficiently? Thank you.
So you're asking about the parallel between the birth of atomic energy and nuclear weapons and the birth of CRISPR and genome editing. They're radically different. Atomic energy and nuclear weapons were government program that was done as part of a national security emergency for military purposes. Genome editing and CRISPR in general is being run mostly out of the private sector and coming from universities. It's being done for peaceful applications for health, for agriculture, and it's also designed to. It's being created an institution, an environment where it is spreading globally without the kind of restrictions we had back in the 1940s and 50s on fissile material and nuclear technology. So they're completely different types of technologies, and the way they've been born is completely different. And to the second point about kind of the misuse, I mean, right now the focus is on prevention of this kind of event. That there's no kind of mechanism or organization to respond to an attack of this sort. But this gets back to the role of something like the Biological Weapons Convention and establishing norms that make even using a genetic weapon to make that unthinkable, to make it not something that maybe countries could do it, they have the capability, but you make it unthinkable. You make it viewed as such a perversion of science that no government, no scientist would be willing to go down that road and use this in any kind of way to cause harm. Right? That's kind of the real goal, is to make it so that all the scientists, all the companies, all the governments are working on this technology, with this technology, they're devoted to peaceful applications of it, and they are aware of the potential risks, and that they're doing things to mitigate them, whether it's a see something, say something, whether it is codes of conduct, it is accepting oversight by regulatory authorities, but really the goal is to have a kind of a common view that this technology has incredible applications for human health and the economy, and we need to safeguard that and develop it for those purposes in ways that it doesn't get derailed or sidetracked by misuse.
Let's just take one or two more questions. We'll take them together if there's more than one.
Hi, I'm Wendy. I'm with a government agency, but I'm asking the question on my own behalf. On page 64, I was surprised to see a recommendation potentially for legislation. Do you have any thoughts about how legislation could be accomplished that would still be adaptive, in David Relman's phraseology, and what committee would be the best one to optimize both the positive benefits and the national security risks?
Let's hold that and see if there's one other question. That was a nice, clear one. Now we'll just go to that one.
Okay, so right, so there was recognition that legislation involves hard law, which can be very rigid and difficult to change, and you're relying on non-experts to craft the language. So the goal would not be to dictate exactly what is, and this is in the context of dual-use research oversight. The goal would not be to dictate the exact nature of what experiments are being judged as being acceptable or unacceptable or needing of oversight. But instead, the core is that right now, the way our dual-use research oversight system operates, it can only oversee research that is federally funded, that organizations or individuals receive federal funding, then they're subject to oversight or to complying with certain rules. Now, the last 2 years, there's been a sea change where now more than 50% of the spending in the US on life sciences research and development is coming from the private sector, not from the government. We've seen in the case of the synthesis of horsepox virus that private funding was responsible for that project. I don't think we yet know where the funding came from for the research by Professor He in China, but to the extent that he did have 2 companies that he was running, maybe to commercialize the technology. Again, you see private funding being an increasingly important source for life sciences research. Our current voluntary kind of system doesn't capture that. So you would need legislation to capture research across the life sciences enterprise, whether it's being done by universities or government labs or in government or in garages or in the private sector. And it would just be mandating that is a national comprehensive system. But I would argue that you'd obviously want the details left to be designed by experts operating on a more flexible basis. But they need to have a legislative authority in order to have that kind of nationwide comprehensive overview.
I think if it's done right, I think that's one of the answers to not patronizing the public. Oh, well, CRISPR is basically always going to be used for good. I think the fact that there are rogue actors it's much more credible to go to the public and say, well, we're proposing, let science do science. However, we're going to also propose some legislative oversight. Because on page, I think it's the graphic on 49, like 2 or 3 of the top risks are people creating things that are worse than anthrax and smallpox. And I think the public likes that people have to get a background check before they get access to anthrax or smallpox, and we know where those labs are inside the US. I'm just talking about the US environment. So I think it's really credible to go back to the public and say, yeah, but if anybody uses CRISPR to make things worse than anthrax or smallpox, we're going to still recommend coverage under a regime, whether it's an existing one or a different one. But so the legislative approach of some kind of oversight where everybody's party to it, I think actually could help strengthen the use of CRISPR and advanced biotechnologies by creating trust with the public.
I think just one other comment sort of coupled to that. A particular interest has been how do you actually, how do you design these types of policies to also be able to have some sort of time scale by which you can revisit, right, the initial assumptions that were laid out in terms of what the appropriate scope of coverage would be, but also critically coupled to that, how do you use those to collect the types of systematic data that you need to inform where the gaps are. So in this case, even the early examples we've seen around trying to do very systematic and rigorous risk assessments have come up to this problem that we actually just don't have the data, not just on incidents, but especially the types of near misses, accidents, sort of behavioral issues. And so it really does require what is often difficult to achieve, but careful policy design that builds in the inherent uncertainties of a new technology. So
let's thank our panel for an excellent presentation. And thanks to all of you for joining us today. Right on time.